Look what I did. Oh, I like you say that. I like soft laugh. It sounds weird. People are like, you freaking oh, sounds good.
Okay, good morning, everybody. Welcome back. It is Tuesday. We have 2 weeks of class left, so we got today and Friday, and then next week, Tuesday and Friday, and then we are done. And these last 4 classes really form a nice coherent unit. So today we're going to do circulation, and then next time we're going to talk about blood and the innate immune system. And a lot of that's going to be talking about different types of blood cells, both in the context of the blood and then also for the immune system, like different types of white blood cells. And then after that, we're going to talk about the adaptive immune system in our second to last topic for the semester. So we'll get into B cells and T cells and how they operate and what they do. And then that will lead us right into our final class, which is on infectious disease. So when we started the semester, if you remember way back in January, we started with viruses and then bacteria. And that's where we're going to end. We're going to end with viruses and bacteria, except we're going to be talking about it not in a biodiversity context or a phylogenetic context, but we're going to be talking about it in the context of human physiology in terms of human disease. So that's where we're headed over the next 2 weeks, and that's going to be it for Gen Bio. That's going to put a nice bow on the whole 2 semesters here. So we're going to get right into circulation today. We're going to start with, as we like to do, a nice big picture of circulatory systems, compare and contrast different types of circulatory systems. We're going to look at that in 2 different ways. There's kind of 2 levels of circulatory systems we're going to compare. Then we're going to look at the human heart, so sequence of HD, human heart function. We're going to see how a heartbeat initiates and how it works. Then we'll talk about the other physical component of the circulatory system, the blood vessels themselves. So sequence the types of blood vessels and the blood flow, and that includes both the pathway through the body and also the what happens at capillaries, how that exchange happens. lymphatic system, which is kind of an accessory circulatory system that does a really important job. We're going to see what that job is up here. So for this section, sequence in HD, the lymphatic system, and compare and contrast the lymphatic system with the blood circulatory system, the main circulatory system. Now the big picture today, sticking with this figure that I love, the big picture today is that we have already talked about a bunch of different ways you exchange with the outside environment. We've talked about digestion, how we absorb the nutrients from the food and get rid of the waste. We've talked about respiration and the different types of respiratory systems you can see. And we've talked about osmoregulation and excretion, how we regulate the contents of those internal fluids. So we've talked about all of these different ways of exchanging with the outside environment, but all of these different methods of exchange occur in very specific locations. But we need to get the results or the products of those exchanges throughout the whole body. And that's where the circulatory system is going to come into play, connecting all of these different exchange structures and systems with each other and with all of the rest of the things in the body, whether it's muscles, organs, whatever it is. You have to connect everything so that you can get all the different materials where they need to be. So that's what we're going to talk about today. How do we accomplish that part of things? Now, when it comes to the types of circulatory systems, as we've seen with lots of other types of systems, some less complex organisms, and we're specifically talking about animals here, so some less complex animals actually don't have a circulatory system. So again, like, we like to pick on jellyfish as like the simple example that don't have a bunch of things, because cnidarians are just 2 layers of cells. So how can they accomplish exchange? No matter what exchange we're talking about, how are they going to do it? They're just going to do diffusion, and they can basically diffuse with the outside environment no matter where you are in that body. You're either going to be facing the outside, or you're going to be facing the gastrovascular cavity to the inside, and that's it. You don't need any specialized structures. But as we've seen over and over again, if you are bigger, if you're more complex, if you're a triple blast with a body cavity, you're going to need some way to connect all of these different structures, and that's where a circulatory system is going to appear. So if you need a circulatory system, all circulatory systems are going to have the same 3 components. The specific structures, the way they work, it's all going to be a little different. But they always are going to have some kind of fluid that's going to circulate through that system. For us, that's going to be blood, but it won't always be blood in the systems we see. You're going to need some vessels that are either going to completely contain that fluid or contain it for part of its trip as it circulates through the body. And you're going to need a structure to pump it. You're going to need a muscular structure, a heart, to move that fluid through the body, whether it's in vessels or not. So all of the circulatory systems we're going to see are going to include these 3 components. Again, the specifics are going to be a little bit different, but they're all going to have fluid, vessels, and a heart. At least one. There are 2 main types of circulatory systems, and it's based on whether the fluid is completely enclosed in the vessels or not. We're going to start with that, and we're going to zoom in on not this kind, the second kind. So the first kind of circulatory system is called an open circulatory system. This is where there is no distinction between the circulatory fluid and just the general interstitial fluid that surrounds all the cells and tissues of the body. So we call that fluid in an open system, we call that hemolymph. And a more general term for that is just your interstitial fluid, the fluid that just surrounds all of your cells. The way an open system works is you have a heart, and then you have some vessels or chambers connected to it that are called interconnected, that are called vessels right here. And then, so the heart is going to take the fluid in, it's going to contract, it's going to push the fluid through those vessels, and then into the chambers of the body. In the body, we're talking about a coelom, right? We're talking about just a body cavity, and it's often going to be multiple different compartments that are all kind of connected together. We call those interconnected sinuses. Sinus just means cavity. So you have a bunch of cavities that are all connected to each other. So the heart pushes the hemolymph and it kind of moves through the vessels and then downwards, it usually kind of filters downwards through the body cavities. And as it does so, it just kind of like washes over all the cells in the body. So anything that's going to be in that fluid that they need, like nutrients from the digestive system, they're going to be exposed, all the cells are going to be exposed to those nutrients, and they can absorb them, and they can also release things like CO2 to get rid of them. Now, as the heart relaxes, it's going to pull the fluid back up. It's like if you've ever used like a little dropper with a bulb on it. Anyone ever used like a little dropper with a bulb? So you press it, and then it pushes the fluid out, and then you relax it. That increases the volume, decreases the pressure. The fluid comes back up. That's how this heart works. It contracts, pushes the fluid outwards and down, and then it relaxes, sucks the fluid back up. And in that way, it circulates the fluid through the body. Part of that circulation occurs within vessels. Mostly, it's just kind of a wash. The cells are just kind of awash in the fluid. Okay, that's an open system. That fluid, again, is called hemolymph. This is what we see in insects. So again, if you take entomology, which is a great class, and I highly recommend you take entomology. It's a really fun class. And if you feel icky about things with lots of legs, you won't by the end of entomology. It's a really fun class. But this is the kind of system you would see if you were to take entomology. Now, an open system is low pressure, because again, it's not enclosed in vessels the whole time, so you have very low pressure fluid circulating through the body. Means it's very low efficiency, and it requires less energy. So this is the kind of thing we see in arthropods, again, like a grasshopper over here. We also see this in most mollusks. This is what you're going to do if you are on the smaller side and you don't have high energetic demands. So like if you're an ectotherm, you can get away with this. If you're an endotherm, if you're going to regulate your body temperature, this is not going to be good enough for you. You need to use the other type of system. Questions about an open system before we get to the other one?
What percentage of the hemolymph stays in the vasculature, and what percentage just like goes into the cavities?
So what percent stays in the vessels versus goes to the cavities? It's ultimately going to be 100% will leave the vessels, because they're just open. It's not like you have like closed tubes and then everything else kind of just around. It's that the vessels are actually open. So basically one complete circuit involves going through the heart, the vessels, and then out of the vessels and back to the heart.
And do the same vessels that release the fluid also uptake it?
I think the heart uptakes it kind of directly. So again, this is the kind of like schematic picture that I love so much. But it's that, so when the heart relaxes, right, it's going to suck the fluid back up in there. It has kind of its own little funnels that lead to it. And then when it contracts, it's going to go through the rest of the vessels that go through the whole body. I can't say specifically for like insects what the situation is, but a lot of times it's like one-way valves. So when you contract the heart, it can't just shoot it back out the same way. It has to go the other direction. So then we have a closed circulatory system. This is your other option. So blood is the circulatory fluid in a closed system, and it is confined to the blood vessel. So obviously, this is what we have. Again, you have a heart, or in some cases multiple hearts, which is the muscular structure that's pumping the blood through the vessels. And those vessels are going to branch as they move through the body. And as you would imagine, ultimately every cell in the body has to be very close to a blood vessel so that you can efficiently do exchange with it via diffusion. If you're too far away, that diffusion is too inefficient. So throughout the entire body, you end up having very small blood vessels. We'll talk about what those are in a little bit. And every cell is going to be within a few cell layers of one of those very small blood vessels. Now this is going to be a higher energy system because it's a constant volume, right? So when that heart pushes on the blood, you decrease the volume of the heart, the rest of the vessels are going to be whatever volume they are. So you're increasing the fluid in those vessels, you're going to increase pressure. It's a high pressure system. That makes it more efficient in terms of the circulation. Means you can get more things like oxygen and nutrients circulated faster. But it also means it takes more energy. Now this is what we find in annelids, so like earthworms right here, very famously. You've probably heard how earthworms, oh, I'm so sorry. You probably heard how earthworms have like 6 hearts or 8 hearts or whatever it is, and it's because at the anterior end, there are muscular blood vessels essentially that all contract kind of in a synchronized way. So there's a bunch of segments of that body near the front where the blood vessels aren't just moving the blood through, they're actually contracting to push the blood through. And you can consider each of those muscular sections a heart. Cephalopods also have closed circulatory systems. That's going to be your squid, octopus, cuttlefish, mollusks, that kind of stuff. And then of course vertebrates, like us, have a closed system, and that's really what we're going to focus on today. We're going to focus on how the vertebrate circulation works for the rest of today. And we'll talk about fish, we'll talk about amphibians, and then we'll get to mammals, and we'll basically just stick with mammals for the rest of the time after that. Questions about closed circulation? All right. So let's talk about how vertebrates do this. The system that is responsible for circulation in invertebrates, we call it the cardiovascular system. Cardio means heart, vascular means blood vessels, so the heart and vessel system. So there's 3 parts to a cardiovascular system. The first is going to be the heart, which again is the muscular organ that pumps the blood. It's responsible for the mechanical force of circulating fluid. Then we're going to have vessels that are going to carry the fluid throughout the body. We'll talk in a little bit about different types of vessels. There's big vessels, there's small vessels, there's different types that are adapted for different things. And then lastly, you need to have very small blood vessels called capillaries where exchange can occur. So you have your heart, you have your blood vessels, and you have exchange in your capillary beds. Those are the 3 components that all vertebrate circulatory systems are going to include. The actual structure of the systems is going to be a little bit different, and there's going to be variations on the same theme, but they're all going to have these things. So we're going to look at a few different vertebrate circulatory systems. This is kind of our second layer of comparison. Right, we can compare and contrast open and closed circulation, and then within closed circulation, we're going to look at the fish, the amphibian, and the mammalian circulatory systems. Now, fish are really unique compared to the other 2 because they have what's called single circulation. So we have a single loop through the body, heart, the rest of the body, back to the heart. It's one continuous loop. That means the heart is going to be simpler than what we're going to see in the other groups. A fish heart only has 2 chambers. It has one atrium, which is where the blood is going to enter, and then one ventricle, which is going to push the blood back out. This system is not very efficient. And if you look at it, you can probably see why that's the case. So what I want you to see here is that you go from the heart, and we're going to go through kind of the pathway for each of these very briefly. We're always going to start with the heart. So you have blood in the ventricle of the heart. It's going to get pumped out. It's going to go to the gills first. Now the gills have a capillary bed because that's where we have to do exchange, right? We have to do gas exchange at the gills. Once you hit the gill capillary bed, once you hit any capillary bed, you're no longer high pressure. You're low pressure. So now you're going to circulate through the rest of the body very slowly. And the only reason you're actually going to circulate is because the heart's going to keep beating and pushing more blood behind you. So it's this really inefficient, slow circuit. You're going to go from the heart to the gills at high pressure, and now you're at low pressure, going through the rest of the body. After the gill capillary beds, it coalesces back into larger blood vessels, and then those are going to lead to additional capillary beds all throughout the body, for the brain, muscles, wherever. The intestines to pick up food. It's more capillary beds anywhere in the body we're going to circulate back to the heart and end up in the atrium, the chamber where you enter the heart. This is inefficient because you spend most of the circuit moving very slowly at low pressure. So it takes more heartbeats to just circulate that total volume of blood through the whole body. So that's how fish do it. That's single circulation. If you're a tetrapod, you do it very differently from this. Now we have double circulation. Double circulation, like amphibians have, involves a heart with 3 chambers, and there are 2 distinct circuits here. So here we have 2 atria, meaning there are 2 ways for blood to get into the heart, and there is a single ventricle which is going to push the blood out. And we have 2 distinct circuits when the blood leaves the heart. It can go one way, it can go the other way, and each of those circuits is a closed loop that leads back to the heart, where you have to get back to the heart before you do the other circuit. Okay, so one of the circuits is called the pulmocutaneous circuit. Let's break that down. Pulmo, lungs. Cutaneous, skin. Because how do amphibians do gas exchange? We talked about this last time, right? They use their skin and they use their lungs. Neither of them are super efficient, but combined they get enough gas exchange to survive. So you have one circuit that's going to lead to capillary beds associated with the skin and the lungs. That's where you're going to pick up your oxygen. And that's this loop right here. So this is your pulmocutaneous loop. And then the second loop is called the systemic circuit. That's where you're going to take that now oxygenated blood and you're going to send it to the rest of the body. So this represents the systemic circuit. So we've now separated the do gas exchange and bring the oxygen where you need it, steps in the pathway to 2 separate circuits. This is more efficient because you can do both circuits at high pressure, meaning you can move more fluid through the system faster. So this is a pretty major advance over what we see in the fish. And just energetically, you can see why this would be the case in terms of their environment. We're on land, we're not in water. Now we have to just spend a whole bunch of energy doing basic things like holding ourselves up. Because we didn't have to do that when we were in the water. We had buoyancy to do that. Now we have to just constantly be using our muscle to hold ourselves up. That takes extra energy. We also are respiring from the atmosphere rather than using our super efficient gills, which means it's a little harder to do gas exchange. We can't do it as efficiently as the fish can, which means we better be more efficient at circulating those gases through the body to compensate. So you can see what the selective pressures would be to be like, you know, if you develop a more efficient circulatory system, you're going to have a big advantage when it comes to competing on land. So there is an obvious downside here. It's right here in the single ventricle. You have deoxygenated blood coming from the body mixing with the oxygenated blood coming from the pulmocutaneous circuit. So the blood that circulates through the body is not 100% oxygenated, and the blood that circulates to the lungs and the skin is not 100% deoxygenated. So it's not as efficient as it could be, but it is better than what we just saw a few minutes ago in the fish. So let's look one more at mammals and birds, because we have double circulation with a 4-chambered heart. And before we go any further, I want to point out that even though the way that mammals and birds do circulation is pretty much identical in terms of like the structure of the heart and the 2 circuits that are involved, it actually evolved independently twice from a reptile ancestor. And that ancestor, we actually see this in many living reptiles, had a partially separated ventricle. It's not, so if you look at like crocodiles, for example, which not coincidentally are the closest living relatives to birds, right? They're the closest relatives to birds and all the extinct non-eating dinosaurs. Look at a crocodile heart. It actually has a partial septum. You don't have to know this. It has a partial septum between the 2 sides of the ventricle, and it basically routes the blood partially separated, and the two kind of halves, the two sides of the blood flow are efficiently kept separate from each other. Birds, and also mammals, have a completely separated ventricle. So we have two atria, one on each side, right and left. We have two ventricles, right and left. So now we have complete separation not just of the circuits, but also of the blood flow in the heart itself. So there's no mixing, which makes this even more efficient. We also have a new name for one of the circuits because we no longer breathe through our skin because we're amniotes, so we can't do that anymore. So we have the pulmonary circuit, which that's heart to lungs and back, pulmonary circuit. And we also have the same systemic circuit, heart to the rest of the body and back. And that's the same as we just saw with the amphibians. These two circuits are completely separate, including within the heart, which makes this the most efficient type of circulatory system. It is not a coincidence that the two groups with the most efficient types of circulatory systems, birds and humans, or mammals, what else do we have in common in addition to our circulation? Is there something else physiological that birds and mammals share that nothing else has? Something with regulation? No? What do we think? Temperature. Birds and mammals are the endotherms. See, you should be confident you had it. Yeah, birds and mammals are the endotherms. It takes a lot of energy to be an endotherm. Right? Think back to last semester, we talked about like the energy transfers with trophic levels and everything. We spend like well over 90% of our energy just maintaining our temperature. That means we have to do a lot more metabolism, which means we generate a lot more CO2. We better be able to get rid of it efficiently and get all that oxygen we need where we need it. It's not a coincidence that the two groups of organisms that have the most efficient circulatory systems are also the two groups that are endothermic. Those two things go hand in hand. Questions about the three types here, the fish, the amphibian, or the mammalian circulatory system? Okay. So we're going to go through the mammalian circulatory system. We're going to sequence mammalian circulation. Don't worry, there are labels on this figure that you don't need to know. Don't worry, it's going to be fine. There are labels on here you don't need to know. Just follow what the pathway I give you. That's the pathway you need to know. We are going to start with the right atrium. Now the heart, again, it's 4 chambers, so it's 2 atria on top, that's where the blood comes in, and then 2 ventricles on the bottom. Those are the parts that push the blood out. When we say right and left, it's within your own body. So like when I'm looking at the screen here, this person is facing outwards towards you. So their right atrium is on your left side, right? So it's relative to within the body we're talking about. So we have the right atrium right here. That's where the blood is going to come in from the systemic circuit. So you've got this picture splits it, so you have like the lower body and the upper body. That blood is deoxygenated, which blue is just universally shown to indicate deoxygenated blood. That blood is going to come in to the right atrium. Now the atria are going to contract, and then the blood is going to get pushed downwards into the right ventricle, right here. When the ventricles contract, the blood in the right ventricle is going to get pushed into the pulmonary arteries. Those are the blood vessels that bring that blood to the lungs. So there's the right lung side, here's the left lung side, we're just following the left side. You're going to go through the capillary beds of the lungs. That's where you're going to pick up oxygen and get rid of CO2. And then you're going to take the pulmonary veins back to the heart and enter the left atrium, now with your fully oxygenated blood. So now again, the atria are going to contract. We're going to push that blood downwards, this time into the left ventricle, right there. And when the left ventricle contracts, that's going to push the blood up into the largest blood vessel in the body, that's called the aorta. It's right here, then it loops and goes down. So this is the aorta right there. And from there, it's going to circulate to all of the capillary beds in the body, drop off the oxygen, pick pick up CO2. And that brings you back to the atrium. After those capillary beds, we're going to take some veins back to the atrium. You don't need to know the names of those veins. So this is the sequence of mammalian circulation. You can see there's some labels I left out here. You don't need to know the labels that I didn't specifically mention. Just like some specific blood vessels, you don't need to know the names of those. It's fine. Questions? Okay. So again, remember when I introduced this, I said we're going to sequence mammalian circulation. That's a bit of a hint, right? We sequence it. That's, you can have an exam question on that. Starting with the blood chamber of the heart, correctly sequenced, right? So, okay, so big picture with circulation, open versus closed, single versus double circuit, 3-chamber heart versus 4-chamber heart, and of course the pathway that blood takes through all of those things. That's what we've covered so far. We're right on schedule. So let's keep going. Now let's talk about the human heart. The human heart is super cool. It's also a little bit terrifying once you know how it works. Because it's one of those like single point of failure things that you talk about in like aviation. Like in terms of aviation safety, it's a thing that you don't want to have anything on an airplane that's a single point of failure, right? You want redundancy, where if something breaks, that's okay because there's something else that will keep it from becoming a larger problem. Yeah, the heart's not like that. The heart is one of those single point of failure things. You need that to work. If it doesn't work, you got a problem. This is a picture from like a 100-year-old med school textbook, and I just really like this picture, which is why it's here. It's hand-drawn, extremely accurate picture of a heart, which I really like. So the heart, we're going to talk about the structure, then we're going to talk about how it works. It is a muscular pumping organ. It has, depending on the species you're looking at, 2 or more chambers that are going to contract, and in doing so, they're going to move blood through the circulatory system. So the atria, the singular is atrium, those are the upper chambers. Those are the chambers that are going to receive the blood from veins. And then the ventricles are the typically larger lower chambers. Those are going to pump the blood out to the arteries. Now the heart is surrounded by this connective tissue sac called the pericardium. And this is important because, the heart, again, is one of those big important things in your body. It's like more important than, your liver, right? Like, if your liver doesn't work, that's a problem, but that's like a long-term problem. If your heart doesn't work, that's a short-term problem right now. So your heart gets a little extra isolation. Not quite as intense as like the blood-brain barrier, but in principle, it's the same idea. You want your heart to be kind of on its own so that things like bacteria, for example, don't have easy access to it. So it's not exactly, it's up here with your lungs in the thoracic cavity. Lungs are obviously a common site of infection, or your trachea or your bronchi, right? These are common sites of bacterial infections. Well, your heart is right in that vicinity, and you don't want the bacteria getting there. So you have an extra kind of layer around your heart that kind of isolates it from the surrounding environment, gives it just a little extra protection, so it doesn't get damaged or infected quite as easily. The heart, in addition to the chambers, includes 4 valves, which are flaps of connective tissue that prevent backflow. So the really important thing with the valves, just in terms of the principle of how they operate, is that when a chamber of the heart contracts, you're able to push the blood through a valve in one direction and one direction only. When a different chamber contracts, that same valve is obviously exposed to the blood flow, right? It came through this door, and then this chamber is going to contract. Well, you can't just go back through that same door. That pressure from the opposite direction actually slams that valve closed. And that's really important to maintain the proper flow through the circulatory system. So there are 2 valves in the middle of the heart that are called the atrioventricular valves, or the right and left AV valves. As you can guess from the name, they're the valves between the atria and the ventricles. So the blood comes into the atria from either circuit, either the systemic circuit or the pulmonary circuit, gets into the atria. The atria contract. When they do that, we'll use So when it contracts, it's really easy to open it downwards, so the blood can flow into the right ventricle. But then when the ventricle contracts, you can see just given the structure of the tissue here, it's going to slam that valve closed, and the blood can't go back up into the atrium. So it's a one-way valve from atrium to ventricle. Does everyone see how that works? Those are your AV valves. So you have a right AV and a left AV valve. And by the way, there are other names for these valves. You don't need to know the other names because they're not as informative in terms of telling you where they are and what they do. I like using the newer terminology for the names of the valves. And then in addition to the AV valves, we have two valves that are called semilunar valves. This was the shape of the valves. They look like half moons. They're called semilunar valves. These are the valves between the right and left ventricles and their respective arteries that lead out of the heart. And again, they are opened by the contraction of the chamber of the heart immediately before them, in this case, the ventricles. So we can keep looking at the right side here. We've got a right ventricle right there. When that muscle contracts, one, it pushes the fluid up. That closes off the right AV valve. It also forces open the right semilunar valve, which is called the pulmonary valve. And I like that word again, the pulmonary valve, because it tells you where it leads. It leads to the pulmonary circuit. Right? It goes to the pulmonary arteries. And then for the left side, kind of tucked in back here, you can kind of see it hiding back there. That's the aortic valve. That's the left semilunar valve. That leads to it tells you, the aorta. And that leads to the systemic circuit through the rest of the body. And then in terms of closing these valves, it's actually blood bouncing backwards, closes it. And you don't have to know the specifics of how this works, but it's pretty straightforward. If you look right here, this is like a little T intersection. So the blood is going to go to the right and the left. Some of it's going to hit the back wall and bounce downwards. And that's going to close that valve. And the aorta does this big U-turn right here. And when it does that, it's under very high pressure. Some of that blood actually kind of shoots up and bounces back down backwards. That closes that valve. Okay, so just like with the AV valves, it's back pressure that forces those valves closed. But the blood, notably, it can't leak back down into the heart. It closes the valves. Okay, so those are the valves of the heart. What questions do you have about valves and how they operate? Now, fun fact, the heartbeat that you hear is not the contractions of the chambers, it's the valves closing. So you're hearing kind of the end of each step. The atria contract, you don't hear that. And then the ventricles contract, and you hear the AV valves closing. And then a beat later, you hear the aortic and the pulmonary valves closing. Those are the two sounds that you hear, and they have slightly different pitches because of the different structures of the valves and the sizes. So the sound is actually the closing of the valves. Fun fact. So now let's talk about how heartbeats work. This is really fun. This is really super cool. Because the heart is not, even though it's muscle, it's not stimulated by motor neurons that tell it to contract. The heart has this characteristic. The heart is autorhythmic. Means it will regulate itself. It will contract or trigger an action potential on a regular kind of pattern on its own, with no input from the central nervous system, no action potentials, no motor neurons needed. So this starts with a structure called the sinoatrial node, or SA node. This is the pacemaker of the heart. This is the thing that sets your heart rate. It's a little mass of tissue in the wall of the right atrium. So right here we have the SA node. Right there. It's just a little bit of tissue embedded in the wall of the right atrium. And this is going to generate an action potential spontaneously. Approximately once per second, a little faster, a little slower, depending on your specific physiology, but 1 to 2 times per second, we're going to generate an action potential within the cardiac muscle via the sinoatrial node. The way this works is super cool. The cells of the SA node are populated with leaky same leaky calcium channels. And the important thing there is they go in opposite direction from the leaky potassium channels. The potassium, because of the gradients established by the sodium-potassium pump, is going to leak outwards. Calcium is going to leak inwards. Now, if you're taking things with a positive charge and moving them from outside to inside, is that going to hyperpolarize your cell or depolarize your cell? What do you think? It's going to depolarize your cell. That's just like sodium moving in, except it's 2+ instead of 1, so it's actually more efficient at depolarizing that cell. This is the initial depolarization step. Think about an action potential. You need an initial depolarization before you hit the threshold. This is it right here. So your heart is just constantly doing that initial depolarization. Then when it hits the threshold, that triggers the heartbeat. We're going to go through all those steps. And then it resets, and the calcium starts to diffuse again, and it triggers the heartbeat. This is what sets the heart rate. And you can increase or decrease your heart rate by increasing or decreasing the rate of calcium diffusion. You can make that membrane more permeable or less permeable to increase or decrease the rate at which action potentials initiate either way. Super cool mechanism. So let's talk about how this works. You take that action potential from the SA node, and it's going to spread through the atria, which makes the atria contract together in a very coordinated way. So think about way back when we did types of muscles. We talked about intercalated discs. Everybody remember those? Those structures between the muscle cells that allow ions to move directly from cell to cell. That's how this is going to work. Once you initiate that action potential in the SA node, it's going to move spontaneously through all of the cells in the atria, causing the atria, right and left, together to contract as a unit from the top to the bottom. And the effect of that is to push the blood downwards into the ventricles. Now once that happens, that action potential is going to reach another node. It's called the AV node, the atrioventricular node. And this is named the atrioventricular node because it is in the wall, or the septum, in between the right atrium and the right ventricle, so the atrioventricular node. This is important because this is another bit of autorhythmic tissue, and it has a really important job. And its job is to pause for about 1/10 of a second. The reason this is important is what moves faster, an action potential or fluid? What's going to move faster? The action potential is going to move a lot faster, right? But you don't want that action potential to move all the way down to the ventricles right away. You need the fluid, the blood, to get there first. So you contract the atria in a very coordinated way. As the blood is moving, you're going to pause that further contraction. You're going to wait for the blood to get down to the ventricles. It only takes a fraction of a second. You just give it that 1/10 of a second pause so the blood moves downwards all the way. Then you can continue the process in the ventricles to pump the blood to either the pulmonary or the systemic circuits. But that pause is really important to make sure that the blood gets all the way down into the ventricles before you contract the ventricles. Otherwise, you'll be contracting half-empty ventricles, and it won't be as efficient. Let's pause here for questions. Let's ourselves take more than a 1/10 of a second pause. Let's see, do we have any questions at this stage? Yeah. That's afterwards, just go to the Purkinje fibers. Yes. Just hold that for just a second, and that's exactly where we're going next. Yes. Okay, let's see any other hands. So yeah, the next thing that's going to happen is we're going to transmit that action potential from the AV node all the way down to the heart apex. It's really important that we're not doing a contraction at this point. So we're going to go down these bundle branches, which are these fibers right here. We're not going to do a heart contraction of the ventricles at this point. Why not? Why do we get down to the bottom before we contract the ventricles? Because where are the valves to exit the ventricles? They are at the top. So if you were to start the contraction right up here, just like you did with the atria, you're going to push the blood downwards. That's a dead end. That won't work. So instead, you basically have cardiac muscle cells that are adapted to just transmit an action potential. It's basically like the heart's version of neurons right here. We call these the bundle branches, or these fibers of these cells that are just for transmission rather than contraction. We're going to transmit that signal down to the heart apex. That's the point, at the bottom of the heart. And then, here you go, we're going to radiate that signal upwards through the walls of the ventricles via these fibers called Purkinje fibers. Now at that point, we can have a wave-like contraction of the ventricles from the bottom upwards, and that's going to push the blood outwards through either the pulmonary valve or the aortic valve. But you need to have that contraction going from bottom to top because of where the valves are that lead out of the heart. What questions do you have about that stage of the process? To get down to the apex and then radiate back upwards, and it's the upwards movement of the action potential that actually triggers the contraction. Yes?
Is it the same action potential that's coming from the SNO, or is
So this is a single action potential that's going to start at the sinoatrial valve. It's going to do the atria and then pause for a second at the the SA node, not valve. And then it's going to do the atria, pause for a second at the AV node, and then transmit downwards and then upwards through either, through one thing. It's not that you have to trigger one and then the other. It's the first one, when it hits that second node, is what causes the action potential in that second node. Yep.
As the SA node, AV node, bundle branches, are they made of the same fibers that the actual cardiac muscle?
So they're all of these cells are modified cardiac muscle cells. So it's the cardiac muscle has basically adapted its own kind of proprietary nervous system, basically. It's not as complex as our, the main central and peripheral nervous system, but it does function as a rudimentary nervous system, where you have a regulatory component and a transmission component to it. And again, the kind of distinction is that this is all muscle tissue, right? It's contractile, but you have a few components of it that are for transmission rather than contraction. Yeah, it's all variations on muscle. It's all adapted from muscle tissue. So here is the whole process. And you can actually see down here the electrocardiogram of what this looks like. So every medical drama, TV show, ever, you always have in the background kind of boop, boop. That's what this is that we just went over. And each part of this process corresponds to a different part of that little graph. So that's our muscle, that's how our heart contraction works. It's a really cool self-regulating process where you're triggering muscle contractions and also transmission in a very coordinated way to move the blood first downwards and then upwards and outwards. I think that wraps up that little bit. So now we'll just talk about how we regulate this. And we regulate this in a couple of different ways. You can regulate the heart rate via the nervous system. And there's a couple different ways to do this. One is with baroreceptors. These are sensory receptors that detect changes in blood pressure. This triangle that's a delta just means change. Baroreceptors are found in the heart and the blood vessels. They detect a change in blood pressure. So for example, if you're sitting or laying down for a long period of time and then you stand up, a little woozy for just a second, it's because your blood pressure went from where it would normally be to you stand up and blood pressure is a little bit lower because now you're vertical. And your baroreceptors are going to detect that. They're going to say, whoa, let's get a little more oxygen circulating, please. And that's going to increase the rate of your blood pressure. This is going to be mediated through the medulla. So you have autonomic nerves that are going to signal the SA node. Again, there's not motor neurons here saying beep, beep, each time telling it to beep. But you have neurons that are affecting the SA node that, for example, can make it more permeable to calcium. It's going to increase the heart rate and therefore circulate more oxygen. So we've talked about things that will affect this before. The sympathetic response, you're going to increase your heart rate and strength of contractions. Parasympathetic, you're going to decrease that heart rate, right? Fight or flight versus can also regulate this. What else? We can do it via the nervous system. What other regulatory means do we have? What else could we use to regulate our heart, do you think? Signals from the nervous system. Any other types of signals that we talked about in the body? What do you think? Nervous system, neurotransmitters, any others? Slower, longer distance? Hormones. We can regulate with hormones. So like when you are under stress, right, the adrenal medulla releases epinephrine. That's going to increase your heart rate. Right? That is hormonal regulation rather than nervous system regulation. Temperature is another one. That leads to hormonal changes that are going to have an effect on your heart rate. So as your temperature goes up, that actually increases your heart rate, which is why a fever leads to a faster pulse. If you ever get sick and you have a fever, feel your heart rate. It'll be faster than normal. When you have a fever, your pulse is just a little bit faster. You can actually detect this at the doctor. They'll check your pulse in addition to your temperature, because those two things will often go hand in hand. And the opposite is also true. Decrease your body temperature, that brings your heart rate down a bit. And these things are mediated through the endocrine system rather than directly through the nervous system. So there's lots of different levers that we can push and pull to regulate the heartbeat. But again, it's all just regulating the rate at which the action potential initiates. Right? How frequently do you hit that threshold? You're not at any point telling the heart, you have to beat right now, now. There's no motor neurons telling the heart when to beat. It's doing that part itself, but there's lots of ways we can regulate kind of around the edges of that to increase or decrease the rate of the heart. So that summarizes the heart and the circulatory system. So again, be able to compare and contrast the types of circulatory systems we looked at and sequence how everything works, right? What's the pathway blood takes, how does the heart work, and put those two together. We can say sequence the pathway blood takes through the mammalian body, starting with the right atrium, and we can have like right atrium, right ventricle, pulmonary circuit, blood, like you do the loop and the heart together. We can include the valves, we can do anything. Just have all those components together as a sequence in your mind. So now we'll talk about blood vessels. And we'll talk not just about the types of blood vessels, we'll also look at what they do, and specifically how exchange works within capillaries. We have three types of vessels we're going to talk about here. We're going to do arteries, veins, and capillaries. We'll start by looking at the structure. Blood vessels can have, they don't have to, but they can have up to 3 layers. You're always going to have an endothelium. That's going to be the layer that lines the lumen. This is going to be very smooth, so that the blood can move very smoothly without impediment through those blood vessels. All types of blood vessels have those. And then the bigger ones, the veins and the arteries, also have the other 2 layers. Smooth muscle, which is going to provide kind of some structural rigidity and also affect the diameter of the blood vessels. We see this much more in arteries compared to veins, because arteries are under high pressure. So you need more muscle to make that structure secure. And then you also have connective tissue. You have lots of elastic fibers and collagen fibers, giving the blood vessels both strength and elasticity. And that elasticity is particularly important in a few of the major arteries, like the aorta and the pulmonary arteries leading out of the heart. Because they're under such high pressure, they do need to flex and stretch a bit to accommodate the fluid at that pressure. Now, when you're looking at the big vessels, the arteries and the veins, the big thick walls prevent diffusion. So you can see here in the artery, you've got a lot of muscle, and then you've got the connective tissue around it. In the vein, it's a bigger diameter, less muscle, you still have the connective tissue surrounding. Even just those couple of layers of connective tissue around the vein, no diffusion from the veins, too thick. So in order to do that, we need to have the thinner ones. So we're going to go through this in the order that blood would go through. And so we're starting from the heart. You're going to go from the heart to somewhere in your body. It could be either circuit. It could be pulmonary or the systemic circuit. It's going to be the same. go into arteries first. Those are the big, thick blood vessels. Those are going to branch into smaller vessels called arterioles that have the same structure, but they're smaller. And then those are going to branch into capillaries. And the important thing with the capillaries is they're the smallest blood vessels, they have the fewest layers. That's where diffusion is going to occur. So we have capillary beds in all of our organs and tissues. They're the things that are going to actually accomplish exchange. So again, arteries, big thick vessels, lots of muscle, connective tissue, no diffusion. You go from there into the capillaries, those are going to be thinner, that's where diffusion is going to occur. Capillaries are basically as small as they can physically be. They are approximately the same diameter as a red blood cell, and in some cases they're actually smaller than that. The red blood cells, they're flexible, so they can kind of squeeze through, but they have to deform and squeeze through a little bit. That's how small the capillaries are. You can also see in this micrograph, compared to what we just saw, that the capillaries have very thin walls. They basically just have that epithelial layer and nothing else. Because this is where exchange is going to occur, this is where diffusion is going to occur, so you can't have a big, thick wall. So capillaries are actually microscopic. And they don't exist kind of on their own as single vessels, they exist in structures called capillary beds. It's just a network of capillaries that are all connected together like a big spider web. Like you can see here how they branch, and they're going to coalesce, they're just going to all connect to each other. This is where the exchange happens between the blood and the interstitial fluid that surrounds all of your cells. So you're going to have gas exchange, and you're also going to exchange the fluid directly, which we'll talk about in just a minute. Well, let's look at these capillaries in a little more detail. We have two very thin layers. Endothelium, which all blood vessels have, and then this other layer called the basal lamina, just kind of holds those capillaries in place, kind of your foundation layer. And that's it. So it's really easy to diffuse. And this is important because this is where exchange happens. This is the only location of exchange between blood and the interstitial fluid. So you need to have your capillaries everywhere in your body. All of your cells need to be near a capillary. Because that's the rule with exchange. It ultimately is going to come down to diffusion, which means that everything has to be close together, because diffusion only works efficiently over extremely short distances. And kind of along with surface area, this is one of our big themes, right? When it comes to diffusion, everything has to be close together. So here we are. After the capillaries do their exchange, they're going to coalesce back into another type of large blood vessel called a vein. Veins are moving from organs to the heart, and it's here I want to make an important distinction. The distinction between arteries and veins is the direction the blood is flowing relative to the heart. If it's going from the heart to a capillary bed, that makes it an artery. If it's going from a capillary bed back to the heart, that makes it a vein. That is independent of the oxygenation state of the blood. Because it's reversed depending on the circuit. For the pulmonary circuit, right, the arteries have the deoxygenated blood. The veins carry the oxygenated blood back. For the systemic circuit, it's the opposite. It's arteries with oxygenated blood, veins with deoxygenated blood, bringing it back. Okay, that doesn't matter in terms of artery versus vein. What matters is the direction relative to the heart. That also means arteries are under high pressure, veins are under low pressure, which means that the structures of the two is very different, as I showed you a few minutes ago. That's the distinction that I want you to have. So you get from your capillary bed, those very small vessels are going to coalesce into venules, which are going to coalesce into large vessels called veins, which will bring that blood back to the heart. Now veins have kind of some important, very specific structural components to them. Because there are two things going on here. One, you're under low pressure. But also, two, for at least some veins, you're moving against gravity. Right? Like if capillary beds all the way down to the toes, So you have veins that are going to run up the length of your legs and your torso back to your heart. Well, that blood is having to move upwards against gravity. So we have a couple of adaptations of the veins to facilitate that blood movement against gravity, even though it's at low pressure after the capillary bed. So one is that the veins are often found kind of in between or surrounded by skeletal muscles. So like as I'm up here walking around, the muscles in my legs, as they contract and relax, are actually pushing the fluid through the veins. It's a different method of moving the fluid compared to like the digestive system, for example, where we talked about peristalsis. That would be way too much energy to move the blood through all your veins via peristalsis. We're not going to do that. It's too much energy, not worth it. So instead, we have the veins run through your skeletal muscles. So just as you move your muscles around, you're pushing the blood physically through those veins. The other thing we find in veins are one-way valves to prevent backflow. So as that blood moves up my legs, if I relax all those muscles, it's not just all going to flow back down to my feet immediately. There's valves periodically, so the blood can only go so far backwards before it stops. Then I contract the muscles some more, and I can continue moving it upwards. This is really important to just kind of keep things circulating. This is why if you've ever been on like a long plane ride, like 5-6 hours or longer, right, intercontinental, you gotta get up and walk around every so often. It's important because if you don't, the blood just kind of pools down your feet. Because if you're not contracting your leg muscles, it's not going to move back up. It's actually those contractions that move it. It's a very important thing to do. So again, I want to emphasize this because it's a very common error. Veins versus arteries, it's all about the direction. Arteries are away. You just think A for away, A for arteries. Veins, I don't have a word that we can use to remember the veins, but if you come up with one, let me know. But veins are back. If you come up with something, let me know. But arteries away. You remember that, you're good. What questions do you have about the structure and function of the three, as we've talked about them so far? Arteries, capillaries, veins. Okay. So now let's talk about the actual mechanism of exchange in a capillary bed. This is really important and really interesting. It leads right into our next part, talking about the lymphatic system. Now, as we said, the capillary has a very thin lining. It's just a single cell layer plus some connective tissue. This allows for very efficient diffusion of all kinds of things: nutrients, ions, gases, waste products. You can move a whole bunch of things back and forth from the interstitial fluid to the blood and the other direction. Now the liquid of the blood, which we're going to talk about next time, blood versus plasma versus serum, we're going to talk about all that stuff next time. But the plasma, the liquid of the blood, is under pressure, right? You can recognize, you can measure this. This is blood pressure. That liquid is under pressure. Some of it actually gets forced out of the capillaries into the surrounding tissues. That's where your interstitial fluid comes from. It's not like a spontaneous thing. It actually comes from your blood. This is why, as we've been talking about, like doing osmoregulation, we regulate the contents of our blood, and that has knock-on effects for all the rest of the fluids in our body. Interstitial fluid, as we'll see in a minute, the lymph in our lymphatic system, and also the cerebrospinal fluid, which is basically the interstitial fluid for your central nervous system. All those other fluids are derived from blood, and this is why. It's because at capillaries, you actually lose some of the fluid to the surrounding tissue. So let's talk about this interstitial fluid. It's a really interesting mix of things going on. There are no red blood cells in the interstitial fluid. Red blood cells can't get out of the capillaries. Small things can get out. Liquid can get out. But cells cannot. Proteins, you have proteins that just circulate in your blood. We're going to talk about some of them next time. Some of those proteins are going to get into the interstitial fluid, but most don't. It has only about 25% of the protein content of your blood, which means the interstitial fluid is hypotonic relative to your blood. Hypotonic relative to the blood in the capillaries. So what other force, in addition to just the physical force of the blood pressure, is going to be moving the liquid back and forth in this system? If we're talking about how one side is hypotonic relative to the other. What's that other word? All the way last semester. I heard it, be confident, nice and loud. Osmosis, that was not nice and loud. It was loud enough for me to hear. It's practically a whisper, that's good enough. Yeah, osmosis, right? There's osmosis going to happen here. It's not just the blood pressure, there's also osmosis. But here's the thing, if the outside, the interstitial fluid is hypotonic relative to the bloodstream, which way is osmosis moving the fluid? It's going to move the fluid, blood to interstitial fluid, or interstitial fluid back to blood? The interstitial fluid is hypotonic. There's less stuff dissolved there. So it's going to go which way? Into the blood or out? If a cell is in a hypotonic environment, which way does the fluid move? It's going to go in, right? That's what happens here. So this is going to be the opposite of the pressure. We have two forces here that are doing opposite things. So we have fluid, not just diffusion, but we also have direct fluid exchange via two antagonistic forces. The blood pressure forces fluid out, then the osmotic pressure moves the water back in because there's fewer solutes dissolved in the interstitial fluid compared to the blood. So this leads to a really interesting dynamic because at the arterial side of the capillary beds, where the capillary beds start, the blood is at high pressure, which means you have a net movement of liquid out. But then as you move through the capillary bed, the pressure goes down. So you stop losing fluid via blood pressure, but that osmotic pressure is still there, moving fluid back in. So by the time you get to the venous end, that's the end that's going to coalesce back into the venules, you're going to have a net movement of fluid in. So as fluid moves through a capillary bed, most of the fluid is going to stay in the capillaries. Some amount is going to go out, and then most of that's going to come back in, but not all of it. It's not symmetrical. Is everybody okay with this before we zoom in a little bit and look at the problem that this causes? Because this does cause a problem for us. It's important for exchange, but the problem is that not all fluid returns. Whatever fraction of the fluid that from the capillaries that you lose, which again, it's not all of it, most of the fluid stays in the capillaries. Some of it gets out, and about 85% of that comes back. Which means 15% of it doesn't come back. Okay, that's fine, that's where the interstitial fluid comes from. But in the long term, this is not sustainable because you're going to have a constant increase in the volume of your interstitial fluid compared to the volume of the fluid in your bloodstream if you don't have a way to recover that blood. So everyone see how that's a problem? Can't have that. You need to maintain a constant volume in your bloodstream, approximately. If you're constantly decreasing the volume of your blood, it's not going to circulate as well, it's not going to work. So we need a solution to this problem. Anyone know the solution to this problem? Hint, if you flip back to the first page of your notes for today, it is the section 4 is the solution to this problem. It's the lymphatic system. That's our recovery system to bring the fluid from the interstitial fluid back into our bloodstream. That's the last thing we're going to cover today, so we'll just briefly do the lymphatic system. Before we get there, do we have any questions about the exchange of the capillaries? Remember, it's via diffusion, and then also directly by fluid exchange. And then in terms of fluid exchange, you have antagonistic forces, blood pressure and osmotic pressure, moving the fluid in opposite directions, but they're not symmetrical, so you lose a little bit of that fluid every circuit, and you have to recover. Questions about any of that? All right. So let's talk about the lymphatic system, our accessory circulatory system. It's not a whole lot we're going to talk about with the lymphatic system. We're just going to kind of briefly look at it in terms of how we recover fluids from the interstitial fluid and get it back to the circulatory system. Then we're going to come back to the lymphatic system briefly next time when we talk about our immune system, because it also plays an important role there. So in terms of the functions we've actually talked about one of these already. Absorb lipids from the small intestine. We did that already. The immune system, we're going to do that next time, so we're not going to worry about that right now. What we're going to talk about right now is circulating fluid from the interstitial fluid back to your blood. That's the only aspect of this we're going to worry about today. And again, one of the other functions we've already covered. The third we're going to do next time after we introduce the immune system. So let's talk about how we circulate our interstitial fluid back to our blood. Is everybody okay with the basic functions? All right, interstitial fluid back to the blood. The lymphatic system is made up of vessels called lymph vessels, and it contains a fluid called lymph, which is very watery, derived from interstitial fluid. So it's not going to take all of the proteins and the solutes that are in the interstitial fluid, but it's going to take some of the water from the interstitial fluid, and it's going to circulate back through the body via these lymph vessels. You can see right here, highlighted this weird neon green. These are the lymph vessels. They coalesce, they meet at crossroads called lymph nodes. So these big bundles right here, those are lymph nodes. Those are going to be important when we talk about the immune system next time. And then in your upper body, you have structures called lymph ducts. Those are the connections between the lymphatic system and the circulatory system, the blood vessels. So that's how the fluid is going to get back into your circulatory system. It's going to be via these lymph ducts. Now the fluid is going to move through the lymph vessels basically like it moves through veins. So lymph vessels are going to be kind of around, or I should say surrounded by, rather, your muscles. As your muscles contract, you're going to push the fluid through your lymphatic system, and ultimately it's going to circulate back into the veins, back into your blood vessels in your upper body. And so that fluid gets returned to your blood vessels. So even though you lose some fraction of your blood each circuit through capillary beds, you're going to gain that back via the lymphatic system, and it ends up being at equilibrium. You're keeping all the different volumes of these different things constant. So the blood, the interstitial fluid, the lymph, you're keeping those volumes constant, even though you're constantly circulating fluid through all three of them. Questions about how the lymphatic system works? Okay. Oh, yeah.
Why lymph as a fluid?
Why do we do that? I don't know. It's just, I think it's just because, so two things. One, it's spatially separated from the other fluids, right? Blood vessel is in, or blood is in your blood vessels. Interstitial fluid is everywhere. And then this other fluid is in this other set of vessels that are physically separated, right? So one, it's in a different location physically. Two, it's chemically distinct, right? It doesn't have as much solute as you get in your interstitial fluid, right? So you have the most stuff dissolved in your blood, then less stuff dissolved in your interstitial fluid, and even less stuff in your lymph. So it's chemically distinct, so we want to distinguish on those grounds. So I was too quick to say I don't know. I think the two answers are going to be, one, it's spatially separated, two, it's chemically distinct. So we call its own thing. All right, so that is all we're doing for today, everybody. That is circulation. We looked at the types of circulatory systems, open versus closed. We looked at how hearts work. We looked at the different types of blood vessels and how exchange works, and we finished by briefly looking at the lymphatic system and how we circulate fluids from the interstitial fluid back to the blood. So as always, we have a practice question for today. You can probably guess what it's going to be. Correct the sequence, human circulation, starting with deoxygenated blood, returning to the heart. And of course, sequence human circulation, right? What organizer would you make to practice this outcome? You would make a
flowchart.
Make a flowchart. Yeah.
5, 4, 3, 1, 2.
Yes. I don't know if anybody else heard that, but yeah. So, want to shout it out with loud, everybody? What's first?
5,
5, 4, 3, 1, 2.
Yes, 5, 4, 3, 1, 2 is the correct answer. Very good, everybody. Okay, so next time we're going to come back. Kind of weird, we talked about circulation. We didn't talk about the stuff that's circulating at all. So we're going to talk about blood next time. And then that's going to lead us right into the immune system, so we'll start that too. Thank you, everybody. Remember, we have our review upstairs in room 205 right after class. See you all.